To this lecture. I'm very pleased to be able to introduce my colleague, Tamas Horvath. Tamas trained in Hungary in veterinary medicine and moved to the US, became head of the Department of Comparative Medicine at Yale University in 2005. His work has focused on the metabolic regulation of appetite and homeostasis more generally, and focuses on a part of the brain which I, for one, have always found very difficult to understand because of the complexity of both neuropeptidergic mechanisms and its lack of simple organizational structure, namely the hypothalamus. When I first learned about appetite metabolism as a medical student, this was really all around insulin signaling, and it was entirely a metabolic problem. And we now know, of course, that a key component of this has been regulation by what happens in the hypothalamus by groups of neurons, the AGRP, POMC sets of neurons. And one of the most, I think, useful things that my reading of Tamas's work has been is to indicate just some of the downstream consequences of those. I point you, for example, to one of his observations about how in the AGRP neurons, when activated in the absence of food, actually lead to repetitive behavior, indicating some of the more complex interactions, which I guess most of us know about but haven't fully taken into account between food, appetite, and behavioral phenotypes. I came across Tamas's work through my own interest in depression and some indications that we'd had of the importance of metabolic regulation through the involvement of mitochondria. And Tamas has made some important observations here, particularly an idea that there may be sets of neurons, again in the hypothalamus, that are responsive to our metabolic state by altering the production of new mitochondria by disruption of he showed this by disruption of the proteins that are involved in controlling the division of mitochondria, the fusion-fission cycle that leads to the creation of more mitochondrial membrane in cells. And then I thought that was my first introduction to this rather unusual way of thinking about the connection between behavior, metabolism, and its central control. So I hope today Tamas will give me more insights into this particularly complicated piece of physiology, and I'd like to welcome him now to give his presentation.
Thank you, Jonathan. Thank you very much for inviting me, and thank you very much for having me. I've been planning this for some time, and today is the day. This is a particular time of our lives, and it's really been an honor and pleasure to be with you through Zoom. I hope all of you are well. I hope all of your friends and family are well, and I hope that they're going to be that way for time to come. I'm going to talk to you about basically my life. I'm going to talk to you about my entire life worth of work that I started back in 1990 when I came to the US from Hungary to Yale, actually, directly, and started to work on the hypothalamus from a different reason. I will mention that for a second, and then ended up working on the same circuits in the last 30 years with different outputs, different behaviors, different autonomic functions in mind. So just before I go to details, this is my lab. Some of them are already gone, some of them are still there. This is, of course, couldn't happen today, but hopefully it's going to happen soon, that we can sit around the table and discuss work and life. My funding predominantly comes right now from National Institute of Aging. Some money comes from the NIDDK, some from NIDA, some from NINDS, and some from Klarman Family Foundation. So I'm going to open up with a sad slide, and this is a slide that shows the main reason why most of us die. These are chronic disorders: heart disease, cancer, respiratory disease, Alzheimer's disease, diabetes, and all of which occur in late onset. So they are in late onset, 5th, 6th, 7th generation of our lives. Unfortunately, there is a new killer, which is COVID-19. But when you actually deal with the real bad case of COVID-19, you will find that also this viral infection impacts predominantly individuals late in their lives with various comorbidities, including diabetes, which I believe will and therefore relate to some of the work that we are doing, and I will show some slides on what we are doing with that particular disease. But in principle, the point is that these are late onset chronic disorders where time is an important component, and actually we are interested in this time component, how this time is incorporated into the etiology of these disorders. And we posit that it's sustaining his workload of the various tissues that on genetic, epigenetic, or environmental vulnerabilities eventually will lead to impairments of various tissues and eventually the demise of the organism. Argument has been made that overnutrition might be relevant for these disorders, including early onset childhood obesity and diabetes, lack of exercise, that these can all lead to propagation of these various disorders. There is no actual real proof that they're connected, but epidemiology and statistics clearly indicate that this may be an important player in the propagation of these disorders. And an argument that this metabolic principle might be relevant for these disorders, if you flip the coin, if you look at the outcome of controlled co-restriction on many issues of cells, tissue, and organism, then you will find that if it's controlled properly, you have decreased cardiovascular impairments, you have lower incidence of cancer, autoimmune disease, neurodegeneration, diabetes can be rapidly diminished with the co-restriction. And you have many, many altered higher brain functions as well. So these two extremes, overnutrition and controlled co-restriction, do argue that indeed there might be a fundamental metabolic principle in the propagation of these chronic disorders. And we do hypothesize that actually this is cellular energy metabolism that provides the time component of disease development. And if we agree to some degree that co-restriction might be beneficial, then what we've been pursuing is that the more we understand about the fundamental principle for co-restriction, the more perhaps we can suggest novel ways of dealing with various diseases. Co-restriction, we can call it a mild form of fasting. These are non-scientific references, but in principle, you move away from glucose utilization more towards fatty acid utilization in this condition. You have changes in your blood regarding many, many factors, including hormones such as ghrelin or corticosteroids or insulin. You can pick your hormone of interest and you'll find that it's going to be affected during co-restriction. You have, you also have alterations, of course, of fuels, lipids, amino acids, and carbohydrates. And one thing that we believe mandatorily will have with this situation is that you will have elevated hunger and appetite. And I will show you through sets of slides why this is, we believe, is a crucial element in this metabolic condition. So, and I show you immediately that answer. In order for you to properly move to a co-restriction state, you will need these neurons in the hypothalamus. These are neurons in the arcuate nucleus of the hypothalamus. They produce neuropeptide Y, they produce agut-related peptide, and they also produce GABA as a main neurotransmitter. They are low in number, a few thousands, depending on the species you are looking at. And they have the same distribution and seemingly function throughout the various species so far studied. So I indicated that I started to work on it 30 years ago. This was my first study that I started when I came to the lab of Csaba Lerant and Fred Naftalin at Yale. We were interested in the relationship between these hypothalamic neurons, neuropeptide Y producing neurons, and another set of neurons in the hypothalamus that produce beta endorphin, pro-opioidal peptides. So it's a set of peptides that are coming out of the POMC gene. And at that time, we were interested in the relationship between these two subsets of neurons in the hypothalamus as it relates to reproductive neuroendocrinology. But in the discussion, we postulated back in 1992 that the interplay between these two subsets of neurons might be relevant for the control of appetite. And that eventually became the case when leptin was discovered by Jeff Friedman. And by many, the function of leptin was connected to the melanocortin system, which is consisting of the AGRP neurons and the PUMS neurons, these MPY producing neurons, and the PUMS neurons in the hypothalamus. Through the works of many, including Roger Cohn and others, and including our own, it turned out that indeed, these hypothalamic neurons, the MPY, AGRP, and the PUMS, are fundamental controllers of appetite, hunger, and satiety. It's a very primitive, simple relationship. The stomach indicates the periphery. When you have low food intake, your various hormones and nutrients are changing in your blood flow. You have elevation in certain hormones, such as ghrelin, that through the circulation signals to the small region of the brain, the hypothalamus, where you have these two substantive neurons, the AGRP neurons, that are activated when you are hungry and promotes all sorts of behavior and autonomic adaptations to that situation. And you have the PUMS neurons that when you take food in and ghrelin subsides and glucose levels elevate in the circulation, AGRP neurons get inhibited. That disinhibits these melanocortin neurons, and activation of that pathway through many downstream targets, including the melanocortin pathway, promotes satiety, and again comes with very specific adaptation and behavior and autonomic regulation. The way the circuitry is set up, which we described back in 1992 in that paper, is that there's a unidirectional relationship between hunger-promoting AGRP neurons and satiety-promoting PUMS neurons, in which these GABAergic AGRP neurons are tonically pushing down the activity of the PUMS system. So the default wiring of the brain is to promote hunger and promote all behaviors that relate to hunger. I'll come back to that later. This scenario is obviously a very reasonable one, because in the absence of such default wiring, you would die after birth. However, understandably, it also becomes a sort of burden when you are in an environment where you don't need to fight for food. Nevertheless, your basic drive is always to pursue food. So this basic wiring is biasing you to always pursue food, even if your homeostatic need is low in pursuing food. There are elements in this circuitry that further complicates your ability to control the output of this system. And that is that this is a very plastic system, even in the adult. So if you're pushing the system towards satiety, it will try to fight back and reorganize connectivity, for example, of the cells, and try to come back to the hunger promoting angle of the machinery. And if you look at inside of this, and when I'm saying that AGRP neurons are promoting hunger and PUMS neurons are promoting satiety, this functionality can actually be completely thrown out, for example, by the utilization of, or the administration of cannabis. We found that if an animal is in a state of satiety, PUMS neurons are firing at high rate. If you expose these animals to cannabis and activate CB1 receptors, then what you will find that very rapidly, the animals are starting to eat ferociously, using the same system as a minute before was promoting satiety is now promoting hunger. And the reason why it's switching to hunger promoting mode is that what they release from the axon terminals, in the case of satiety, this is alpha melanocyte stimulating hormone, which acts through the melanocortin 4 receptor to promote satiety. If you bring cannabis to the system through a mitochondrial mediation, the pool of vesicles that were releasing MSH are not released anymore, and they switching to another product of the PUMS gene, which is beta endorphin, and beta endorphin will be released in response to cannabis, and through opioid receptors, they're going to be generating a very high interest in eating and promote all the behaviors associated with eating. So again, it's a very flexible system at every level, including the mitochondria. So inside of the cell, when cells are moving towards AGRP neurons are moving towards activation and promotion of feeding, they go through, the mitochondria go through fission. This fission mechanism is driven by very specific lipid metabolism pathways. On the other hand, the neurons, the pumping units, when you eat and glucose levels elevate, mitochondria goes through fusion and tethers to the ER, and this is an important component element in the activation of these neurons to be able to promote satiety. We have been conducting other studies as well, and finding that this fission and fusion of the mitochondria is relevant to other cellular functions as well, or the functionality of other cells as well. That includes T cells, that includes any sort of cells that are responsive to the changing metabolic milieu. An important element in the fission process driven by lipid metabolism, that it is a process in which there is a very strong control of reactive oxygen species production. So they keeping relatively low the level of ROS while the cells are being activated, versus when you load glucose to these cells, you promote fusion of the mitochondria. ROS is generated at a much higher level. In fact, ROS becomes a very important signaling molecule in these systems to promote the proper functioning of that cell in that given moment. Now, this dichotomy between activation of hunger-promoting neurons and in general intracellular events associated with hunger versus those associated with satiety may also explain why it's so difficult to find a mechanism or a approach for example, to fight obesity, to promote satiety, because when you promote satiety, you will by default activate these neurons and basically promote glucose utilization, and you will be successful in activating these cells. But what will occur in the long run chronically is that this elevated glucose level that is not only present in these cells but also systemically will induce massive impairment of cellular functions through ROS, and eventually these cells can die. In fact, the whole organism can die. So it's a sort of a catch-22 when it comes to promotion of satiety and weight control. But we took and exploited these mitochondrial events to study other cells and other tissues and other events as well. For example, we showed a few years ago that Zika virus infection of cells in the brain really impact the mitochondrial function. We are now finding that SARS-CoV-2 is also impacting very strongly mitochondrial and cellular metabolism. So these principles that we uncovered regarding the hypothalamic feeding machinery is relevant to other systems and other pathologies and physiology as well. So just going back to the topic of my talk, hunger, and I'm going to be talking about behaviors, how these neurons in the hypothalamus impact other behaviors than feeding, as Jonathan alluded to. So when you think about hunger, obviously, besides eating and getting fat, you will find that all the behaviors that we have are connected to hunger and satiety. Simple ones, you need to be awake. So we suggest that those parts of the brain that are important regulators of sleep and wakefulness need to be connected to these hunger controlling neurons. And when you have disorders associated with them, that these neurons might be playing a role as well. You need to remember and locate food. So we argue that short and long-term memory and related circuits are connected to these neurons. And diseases that emerge in association with the impaired function of these areas may also be affected by these cells. Decision making. So any behavior that you pick, we argue that you will have an impact from these hypothalamic neurons, understandably, because again, when you're hungry and you need to pursue your food, you need to have an adaptive, complex adaptation to that, which will have to involve all sorts of behaviors. So then a simple question is how critical AGRP neurons are for life, which is kind of the title of my talk and what I'm trying to convince you that they are important. We have accidentally, we and Richard Palmiter's lab accidentally addressed that question a few years ago, 15 years ago. Back in 2005, due to the great work of Richard Palmiter, we sort of suspected that these neurons are important for feeding, but when Richard knocked out AGRP, knocked out MPY, he didn't find that these cells or these systems are so important for regulation of feeding. So then a new technique became available where you could tag these neurons genetically with diphtheria toxin receptor. So his lab and my lab in collaboration with Jens Brüning's lab in Cologne developed this methodology and killed AGRP neurons with the administration of diphtheria toxin in adult animals. And what we found was that indeed, if you eliminate these neurons, which can occur within a few days, animals were eating less and less, and they were losing weight, which really was this was the key finding of these two studies. And these were the first experimental studies to actually prove that these neurons are fundamental for feeding. But what was the outcome of this intervention? That all these animals died. Now, you may say that, of course, if you don't eat, eventually you need to die. But we were sort of asking that question more philosophically, and that is that if you have this very complex cortical regions and other regions of the brain over evolution, and you have this primitive subset of neurons, you would have could have anticipated that that sophisticated component could override the loss of these cells, which I come back to that it actually can happen. The alternative take on this was, which we've been pursuing, is that perhaps the role of these primitive AGRP neurons are more crucial, more fundamental for the coordination or control of the complex behaviors than previously thought. So that actually you can overcome the loss of these cells is also shown in Richard Palmiter's work, where he finds that if you kill these neurons with this diphtheria toxin receptor approach early after birth, then actually the animals will survive that. They grow up and they become not relatively, but predominantly normal. I'll come back to that issue later. So if you do it in a time when there is more flexibility of the brain, if you want to put it that way, then they're fine. But if you do it in the adults, then these animals will die. Now, there is a condition which we believe is very similar in human disease to this situation, which is cancer cachexia. And if you look at the phenotype of these animals, they're very similar to the progression of cancer cachexia in that eventually you lose the ability to swallow. And eventually the reason why you die is that your diaphragm becomes digested and your breathing collapses. So you may say that when you kill cells in such a dramatic fashion, few days in the hypothalamus or anywhere else in the brain for that matter, that you might be inducing a local infection, which may lead to a sort of a sickness behavior. That's why the animals are not eating. So with Jens, we then took the other cells out, the POMC neurons that promote satiety, with the same approach, and found that actually these animals were gaining weight in line with the notion that if you remove satiety, animals would be gaining weight, and they were eating more. So it proved the point that these POMC neurons are important for satiety, and it also showed that this is not simply due to the loss of cells that causes local infection, that we change the behavior outcome of these animals. So then again, our question had been to what extent these AGRP neurons might be relevant to other behaviors than hunger per se or feeding per se. And Marcello in my lab at that point started to interrogate these questions by generating various models. In this paper, he summarized his results on these animals where he selectively expressed TRPV1 channels in the hypothalamus, in the AGRP neuron specifically, in a TRPV1 whole body knockout background. So now capsaicin will selectively affect these cells only in the body. And what he found was that if you injected capsaicin, if you were recording from a slice, then you induced action potential generations in the AGRP neurons. If you administered capsaicin to living, live animals, then you found that there is a robust induction of feeding evoked by this injection, which mimics optogenic activation of these cells. In fact, the onset is faster than if you were to do optogenic approach, showing that indeed the model is working. And if you look at these outcomes on the freely moving animals, then you will see that they going to be ferociously eating for a prolonged period of time after administration of capsaicin. So now he took these animals and eliminated food from the cage and looked at various behaviors in various settings, open field, zero maze, marble burying, and found that if you activate these AGRP neurons, they do go to the center to explore the novelty, but if you activate the AGRP neurons, they're more inclined to explore the novel object in the center of the cage. If you put the animals in a zero maze, you will find that most of the time they prefer to be staying in the closed arms. When you inject capsaicin, then you will find that they start to be moving out to the open arms, and eventually they would be spending more time in the open arm than in the closed arm, suggesting that AGRP neurons are driving these animals to go against fear and or work against anxiety or suppress anxiety, whichever way we can interpret this outcome. And he set up this little paradigm where he puts marble pieces into a cage, places the animals, and these animals like to dig and bury these marbles. When he placed the animal after administration of capsaicin to activate AGRP neurons, these animals became much more obsessively burying these marbles, and no matter how many marbles you would give to these animals, they would keep on burying. If you set a time, 5 minutes, 10 minutes, then you will find that vehicle injected animals would bury 30% of the marbles. These guys with activated AGRP neurons would bury much more. But again, if you were to maintain this, they would bury as many marbles as you provide for these animals, suggesting that there is a anxiety and increased compulsion associated with AGRP neuron activation in the absence of food provided. If you were to provide food, the animals would go and pursue feeding. So acute activation increased interest in novelty, increased drive to overcome anxiety, and increases the animals to engage in compulsive behaviors. We also showed that if you do fMRI, but obviously from the behaviors, you can predict that there's a massive and rapid shift in cortical and subcortical activity of these animals, which is diffuse. There is no very specific aspects of it, and this will, I will come back to that later. So when we saw these sort of outcomes in association with the AGRP neurons, we were wondering whether perhaps when you have mental alertness, altered mental alertness in certain situations, such as schizophrenia and anorexia nervosa, these neurons might be relevant for the propagation of those manifestations. And then we had started to work and conceptualize these disorders from a metabolic perspective. And we're basically looking at, in this case, the schizophrenia, the history of schizophrenia, how it was pursued. And, you know, if we looked at the old work of insulin-induced coma, starting in the 1920s, and it was clear that those people who, with the positive symptoms of schizophrenia, who were exposed to insulin-induced coma, came back and survived, which obviously is a key aspect of it, and eventually was abandoned because of the high mortality rate. But if you survive that intervention, then for a prolonged period of time, you would have no positive symptoms of schizophrenia. Of course, argument can be made that that is basically a sort of a complete turning on and off of a computer kind of thing, and that may have nothing to do with metabolism, despite of the fact that, of course, insulin is knowingly a key regulator of glucose utilization. If you were looking at certain and many of the atypical psychotropic drugs that have been effective in treating positive symptoms, they cause many, if not most of them, cause massive obesity and diabetes, suggesting that these two together, for example, that indeed fuel utilization might be an important impairment in certain circuits that underlie the emergence of those symptoms. And coming from a completely non-scientific take on that, and that is that if you are you're dreaming, you're asleep, you're having those kind of things that you would associate with positive symptoms of schizophrenia in an awake individual are acceptable to occur during your dream phase. Again, we look at it from the perspective of a major difference in systemic metabolism during sleep and wakefulness, more towards fatty acid utilization in sleep and more glucose utilization in awake state. Again, these are non-scientific takes, but aided our conceptualization that perhaps these neurons that we are dealing with that are fundamentally important for systemic metabolism control might be relevant for in the etiology of these kind of disorders. So obviously there's no avoidance to argue that the cortex and other structures are fundamentally important for those phenotypes, but metabolic principles might be relevant, and those then could be driven by hypothalamic neurons. Both anorexia nervosa and schizophrenia have major developmental components, specifically related to peri-adolescence, and then through a set of studies, again with the lead of Marcelo in my lab, we're trying to look at whether AGRP neurons have some relevance for development of circuits and development of behaviors. Marcelo then went and took an advantage of an animal that we generated some time ago. In that we basically exploited our understanding of the signaling pathway within the AGRP neurons that enables these neurons to fire when you are hungry. And one of those molecules that are important for this switch was SIRT2-in-1. So Marcelo generated animals in which he removed SIRT2-in selectively from AGRP neurons. And what happens is, in this case, you record from slice, and the AGRP neurons had certain frequency of action potentials. You add ghrelin to these preparation. Ghrelin is the hormonal signal that comes from the periphery to signal that you are hungry, you need to eat. And you will find that these neurons are starting to be firing at a higher rate. When you remove SIRT1, these remain to be neurons, they fire, but they don't sense properly, if at all, these signals coming from the periphery to argue that you should be increasing the firing and increase your eating. So what is the outcome of this intervention on non-food related motivated behaviors? We found that they actually eat less, so that component was fine. And this study showed that in these animals in which you have impaired AGRP neurons from birth and from, this germline thing, and they have lower interest in food, they have increased interest in novelty, for example, which was in contrast to what we found in the adult animals. In the adult animals, we found that there was a positive correlation between the activation of these neurons, AGRP neurons, in a way activation in between actual hunger and complex behaviors, including novelty. Here, when we interfered with these neurons through development, you had lower interest in food, but higher interest in novelty. Just show you the quantification, which then immediately suggested to us that perhaps there may be an impact of these neurons during development on VTA dopamine neurons. In fact, that's what we found. So when we were recording from midbrain dopamine neurons of these animals in which AGRP neurons were selectively impaired, there was a much higher excitability of these dopamine neurons, in this case, in the propagation of LTP in the absence of any cue. And there was increased amount of dopamine in target regions of the ventral striatum, in this case, of the VTA dopamine, in this case, the ventral striatum. So we had this situation where we altered AGRP neuronal function from conception, if you want to put it that way, which then resulted in an altered organization and activity of midbrain dopamine neurons. To sort of strengthen that notion, if we were exposing these animals to cocaine, we found that after injection of increased amount of cocaine, the locomotive response was significantly elevated in those animals where we have impaired AGRP function relative to controls. when we set up a condition place preference arrangement here, also while both the both the knockout and the control animals had elevated preference to the region where they received the cocaine, those who had impaired HGRP function, they have more preference to that side. Just showing that indeed selective alteration of HGRP function impacted the functionality of the adult dopamine system. VPA projects to other sites as well, such as the prefrontal cortex. So we start to be interrogating the same simple questions. Do they impact prefrontal cortex? Do they impact dopamine system? And how that comes about on function or structure and function of the prefrontal cortex. This was done originally by Mike Watterson, who since left my lab. So we confirmed that similar to the ventral striatum, the output of the midbrain dopamine system to the prefrontal cortex was also elevated. So you had elevated dopamine levels. What we found here was that by the time of peripuberty, post-puberty, early post-puberty, there was an overall decrease of cell number in the prefrontal cortex of these animals. And this lower number was included neurons, so lower number of neurons, lower number of microglia, as well as lower number of astrocytes. So it seemed that there is a robust and significant impact of altered HGRP function, we assume, through altered dopamine levels on the development of prefrontal cortex. And when we exposed these animals to this paradigm of pre-pulse inhibition, we found that actually those animals who had impaired HGRP function had impaired ability to adapt to a pre-pulse in these chambers. We found that a non-selective receptor antagonist reverted the loss of cells in the prefrontal cortex when administered in that postnatal peri-adolescence period, and also the impaired pre-pulse response of these animals, suggesting that indeed these neurotransmitters are relevant for the proper development of the prefrontal cortex and potentially, of course, other regions of the brain as well. So impaired HGRP neurofunction during peri-adolescence decreased prefrontal cortex cellular integrity and resulted in behavioral alterations. And I'm very careful in saying, but I say that to make my point that is moving the animals towards more like a schizophrenic type of state when it comes to, at least when it comes to pre-pulse inhibition. So we also been pursuing anorexia nervosa, how these neurons might be relevant for manifestation of symptomatology characteristic of anorexia nervosa. And these studies really come from the idea that in order for you to develop anorexia nervosa, you need to diet. And of course, many of us diet, and very few of us end up in this situation. And based on the literature, it's emerging that in order for this to really be propagated most effectively, this disorder, you need to have the constellation of diet, social stress, and exercise in a very specific period of time, which is, again, peri-adolescence. So in this study, this was Maria Miletta in my lab, she pursued these questions by interfering with HGRP function, a very specific paradigm of anorexia nervosa. It's activity-based anorexia nervosa paradigm. Here, animals are food restricted, so you need to have a diet component. You food restrict these animals to 2 hours of ad libitum, 2 hours feeding during dark phase, and you provide a running wheel for these animals. We had 2 sets of animals. In one set, we elevated HGRP activity using the capsaicin model, the TRPV1 channel model, and analyzed how these animals are performing at a time when you food restrict them and you make the animals the running wheel available. And you will find that control animals start to run, and eventually they have a decreased ability to run, versus when you activate HGRP neurons, they capable of continuously running for prolonged period of time. And during this 72 hours of testing, you find that no control or no AGRP activated animals die despite of the fact that they actually run much more. When we impair AGRP neuronal activity, then what happens is that these animals start to run, but very rapidly they fall off the wheel, and eventually they die. So all the animals in which we impaired AGRP function by removing these cells from early on, this is the approach that Richard Parmiter showed, that you can eliminate AGRP neurons perinatally and the animals don't die. So they have alternative ways of promoting feeding. But when you remove these AGRP neurons and you expose them to this anorexia nervosa paradigm, that all of these animals die. So what we looked at in these animals, whether they might be differential metabolites from the periphery available during the time of running. We did that because we know that the hypothalamus, these neurons and other neurons, are very much involved with autonomic control of peripheral tissues, such as the fat depots. When you don't eat, when you diet or you fast, you start to be using alternative fuels from the fat. That needs to get a signal, and that signal is coming from the brain and in part coming from these hypothalamic neurons. And what we found was that those animals that we had impaired AGRP function and they were running, they were not able to mobilize the fat in the periphery, and fat was just going down and down. And we hypothesized that this is lack of fuel that drives these animals to have impaired ability to run and eventually to expire and die. And in fact, when we replace the fat in these animals with a high fat diet, so we food restricted these animals again, but that limited amount of food that we gave now was in the form of high fat instead of the standard chow that has limited amount of fat. And what we found was that when we exposed these animals to the same paradigm, they were capable of running relatively well, but more importantly, there was no death associated with this scenario. So arguing that indeed, this is the fuel mobilization from the periphery that plays a very important role here for both enabling the animal to run under these conditions and to survive and not to die. I forgot to mention that anorexia nervosa is one of those psychiatric conditions with the highest mortality. So this mortality, we suggest, might be relevant, or might be related to altered AGRP function. So peri-adolescent interference with AGRP functions coupled with food restrictions and compulsive exercise resulted in phenotypic alterations characteristic of anorexia nervosa, and that the alteration of all these events are to some degree related to great degree, we believe, is related to fuel mobilization, and that is driven by the AGRP neurons. So overall, we suggest that AGRP neurons may play a fundamental and previously unappreciated role in normal development and control of complex behaviors, both via ascending pathways. In this case, I showed you an example of dopamine, but we argue and suggest that there may be other ascending pathways as well as by control of peripheral fuel mobilization. So this is the reason why we suggest that these neurons have relevance to many dysfunctions of the brain related to all sorts of behaviors, including schizophrenia and anorexia nervosa. And again, that this is mediated both by pathways directly going up towards cortical regions, as well as pathways through the autonomic nervous system or the endocrine hypothalamus that will impact peripheral tissue function, and together these will have an impact on how a given cortical region or region of importance for a behavior is functioning under different circumstances. So just to finish with the overall question of survival, to what extent alteration of AGRP function determines overall survival of animals. We have been looking at various tissues. As I told you, we showed that intercellular events in fat tissues are altered in response to altered AGRP function in the hypothalamus. In this case, we looked at Beijing. We showed that depending on the activity level of the AGRP neurons, you have different level of bone metabolism, higher AGRP function promotes increased bone mass, and decreased AGRP function promotes decreased bone mass. We also showed that activity of these AGRP neurons have an impact on how peripheral tissue T cells function, effector and regulatory T cell communications are affected, with implications of many things, including multiple sclerosis and aging in general. And when we set up these cohorts of impaired AGRP functioning animals, either knocking out the SIRT one as I showed you before, or knocking out a mitochondrial transcription factor from AGRP neurons, which eliminates these cells from the hypothalamus in a few months. We followed these animals. We looked at what these animals die of during the course of the study, which predominantly B-cell lymphomas, what they die of. Interestingly, we found that actually the cancer propagation in these animals, the knockout animals, is significantly diminished compared to control animals. And you have a situation where you have diminished cancer propagation, lower interest in food, increased physical activity, increased interest in novelty, enhanced adaptive immune response, and overall decreased disease burden. Nevertheless, we found that males specifically had a short lifespan when they were provided ad libitum feeding. So despite of all these correlates of supposedly healthy aging, these animals died earlier, the males died earlier than the control parts in both of these cohorts. Females did not show the same response to this. Again, this is ad libitum feeding. We are right now running studies to see whether the co-restriction associated prolongation of lifespan might be affected by these functionality of AGRP neurons. So just to summarize, some cliches here: the integrative physiology relies on proper communications between various tissues. The hypothalamus actually is in a great geographical position to enable the proper alignment of peripheral tissues with behavior in particular. We also suggest that one tissue phenotype in a given vulnerability of disease may predict the potential vulnerability of another tissue, and also that behavior phenotype may predict vulnerability of peripheral tissue phenotypes to various diseases. And basically, that's where I would like to stop, and thank you for your attention.
Thank you, Tamas. So what facility do we have for asking questions?